All right, welcome, welcome. So today we're talking about regulatory genomics. So this is the basis of gene regulation. This is sort of how our cells basically decide what to do. So we're going to start with an introduction to the computational problem by setting up sort of what are the biological parameters, what are the biological data sets, and what is the computational problem we're trying to solve. Then we're going to look at some classical approaches for solving this problem. And then we're going to sort of dive into how are all of the tools that we've learned so far, including convolutional neural networks and recurrent neural networks, allow us to address this problem using deep learning. So what are the basics of gene regulation? So as you all know, you have one genome within you. You have your genome, 2 copies, one from mom, one from dad, mixed and matched in very cool ways. But you have many, many, many cell types. If you look at the diversity of cell types within, any organ of your body, like within your blood, there's dozens of different types of immune and non-immune cell types. Within, just your skin, there's just all these different cell types that are sort of extremely specialized. Within your brain, within, your heart, your lung, your liver, the iris of your eye, the hair. It's incredible how much diversity you have in function, even though they're all running the same code, right? I challenge you to write an automaton, which basically has one piece of code that starts out as a zygote and then divides and divides and divides and divides. With all of the tools of sort of modern computer science and robotics and automation, etc., we still can't build anything even remotely to the complexity of this cell autonomous program that will basically, grow up to become a fully functional human being, where the blood actually flows through the veins and doesn't just like diffuse in random places. And, the electrical signals start from one neuron and make it all the way to the other neuron, where, despite all the visual and auditory illusions that we saw in the last couple of lectures, you're still able to sort of understand and make sense of the world around you. So I want you to pause for a split second and reflect upon the complexity of that problem. You're not starting with like 20 different, specialized engines for every one of your cells. No, you're starting with one single code. Okay, so the challenge that we're going to see today is how does the body actually solve that? And then we're going to look at sort of how to computationally sort of infer the structures that the body is using. So the first thing to note is that the way that this is solved is in part due to the packaging of the DNA. So every single one of your cells has a nucleus, or most of them, except for, I don't know, the one actually shown here. That's your red blood cell. They get rid of their nucleus to sort of fit more hemoglobin and more oxygen pumping through your blood. But anyway, nearly all of your cells have basically a nucleus. And inside the nucleus lie 23 pairs of chromosomes. And these chromosomes are not just like floating around like spaghetti. They're packaged up extremely tightly into really tight spaghetti that we call chromosomes. Very creatively named. They're colored things. Like under the microscope, you would basically start seeing these colored things, chromo, which means color, and soma, which means body, colored body. So colored stuff. So these chromosomes are basically where the DNA sits. And we all like to sort of draw chromosomes like that. So in preschool, that's how you learn how to draw chromosomes. But this is the very simplified view, right? By the time you hit primary school, you sort of learn that chromosomes spend only a tiny amount of fraction in that form. Most of their time, they're actually being read up and used up, and they only get nicely, tightly packaged right before cell division. So what do they actually look like inside the cell? Well, frankly, we have no idea, right? At this scale, we know that DNA looks like a double helix, right? The most noble molecule of our time. This basis of heredity, the fact that every single strand can serve as the template for the complementary strand. And yeah, when my kids turned about 4, they were able to replicate DNA on the fridge wall by sort of writing a T every time you see an A and writing a G when you see a C, right? It's stuff that like kids can do. But then beyond that and below that, this has been a mystery for many, many years. And it's only in the last few years that we basically realized what's going on in between. First, with the advent of bigger and bigger chromosomes, we saw beads on a string. Turns out those Bs were nucleosomes. Every single strand of DNA is held together by these nucleosomes. So they wrap around these nucleosomes every 147 bases and about a 50 base pair linker. So you can think of the genome as basically 200 chunks, 200 base pair chunks, right? Just going on for a long time. And now each of those chunks is then packaged up into higher order structures known as chromatin, very creatively named after chromosomes. And then this chromatin is looping and looping and looping in structures that have only in the last few years been visible through 3D folding techniques that we're going to cover a lot more in future lectures. And eventually you get to the DNA. Who feels that they're learning stuff already? Yeah, good, awesome. So that's part of the way that your cells have different identity. Even though they all have the same code, their genome is the same, but their epigenome is different. So epi means on top of, genome means genome, so on top of the genome. So there are a series of modifications that can basically tell the DNA to package itself differently in every one of your cells. Everybody with me so far? And then these modifications are basically enabling us to now learn about different classes of elements in the human, in the DNA of every one of our cells. Okay, what are these different types of modifications? The first one is on the nucleosomes themselves. Nucleosomes come in many different flavors. There's some green ones, there's some yellow ones, there's some red ones that taste like strawberry, and so on and so forth. So these flavors of chromatin and these colors of chromatin, whichever way you want to call it, these states that chromatin assumes in different cell types, is basically encoded in post-translational modifications of the histone proteins that make up these nucleosomes. Okay, let me unpackage all that. Okay, so there are 8 histone proteins inside each octamer, from the Greek 8 parts, octamer, which is every nucleosome. So every nucleosome is an octamer. There's 2 H2A, 2 H2B, 2 H3, and 2 H4 histone proteins, okay? And every one of those histone proteins has a very long amino acid tail that sticks out, just like that. And in that amino acid tail, you can encode information on the packaging. Kind of like, I don't know, in an Amazon warehouse, every box has a tag of where it should be going and what it should be doing and when you should be picking it up, okay? So in the same way, the packaged up chromatin has tags that say, oh, open me up if you're a neuron, open me up if you're a liver cell. And the open me up is placed in different locations depending on whether you're a liver cell or a brain cell and so on and so forth. Is everybody with me so far? So this is basically at the foundation of epigenomics. And that's at the foundation of how we end up with the diversity of cell types that we have in the human body. And that's one type of modification which can assume hundreds of different individual marks and countless combinations of those marks, okay? So we're gonna call them H3, K4, ME3. What does that mean? That means that it's histone protein H3. Remember H2A, H2B, H3, H4? Again, these are sort of parts of the nucleosome. So on histone H3, on lysine 4, so K stands for lysine, there are 3 methyl modifications, 3 methyl groups, CH3, CH3, CH3. Everybody with me so far? So that's what these cryptic names mean. And their colors is all you need to remember. H3K4 trimethylation is usually associated with promoter regions that are red. H3K4 monomethylation associated with enhancer regions that we're going to call our yellow or orange. H3K36 trimethylation associated with green regions, which we're going to call transcribed regions. H3K27 trimethylation, repressed regions, which we're going to call our gray. Okay? Is everybody with me on sort of basics of gene regulation? So basically, one genome, many, many functions. The way we encode the functions is by differently packaging chromatin to keep open the genes that we're going to need in every one of the cell types. And that packaging is encoded through epigenomic information. One type of epigenomic information is histone modifications. Another type of epigenomic, and there are many different classes of chromatin. There's many different flavors, many different colors. That's one type of epigenomic modification. And another type is the accessibility of DNA. Basically, if DNA is packaged up very tightly in condensed chromatin, transcription factors or regulators cannot bind there to turn genes on and off. So these regions are usually repressed. We call them heterochromatic, as opposed to euchromatic, which are open. Eu in Greek means good, like a eulogy or euthanasia. I don't know, killing well, sorry. Usually eulogies come after euthanasia. Anyway, and then the third type of modification is at the DNA itself. So CpG dinucleotide can have a methyl group directly on the C with CH3 only. And then this is sometimes called the fifth nucleotide, even though there's many different types of modifications, both at the DNA level and at the RNA level. Okay, so CpG methylation or DNA methylation is usually a repressive mark in regulatory regions. DNA accessibility is a mark of access, of openness, of open for business kind of thing. And then all of these different other colors and flavors are associated with different classes of elements. Raise your hands if you're with me. Awesome, good. Raise your hands if you've learned something so far. Nice, awesome. Raise your hands if you knew everything already. Okay, good, good. Yeah. Louder.
These modifications, basically the last one you mentioned, so these are like inherent throughout the replication. It's not replacing this.
What a great question. The post-replication preservation of epigenetic information is a major area of research. Very often you will ask a question and I will say, oh yeah, here's the answer. Other times I will say, I don't know the answer. And the third class of possibilities is no one knows the answer. So I wouldn't say that no one knows the answer, but we're still figuring out the answer. And in particular, there's a lot of work about epigenetic inheritance. Namely, hey, if this information can survive a cell division, maybe if I eat a ginormous burger today, my children in 2 generations are going to be obese. I mean, I'm exaggerating things a little bit, but basically maybe the decisions that you make in your lifetime, kind of like stretching my neck to reach for an apple, might have impact on offspring. For example, giraffes having really tall necks, right? This was all dismissed as Lamarckian evolution and BS when Darwin's theory of evolution came about, even though if you read closely, Darwin actually believed in Lamarckian evolution despite what you've read in the popular press. So suddenly Lamarckism is back with epigenetics. Everyone's like, ooh, that means that if I stretch my neck, maybe my offspring will have longer necks. Not as simple, way more complicated. Why? Because as soon as I produce sperm or my wife produces eggs, there's a round of reprogramming that wipes out epigenomic information. Then as soon as the two mix together to make one of our children, there's another round of wiping out that epigenomic information. So the genome goes out of its way to wipe all that information clean across generations. Now within the body, if I have a liver cell and it divides, is it now like, ooh, what am I? Am I a brain cell? Not really. Some of these processes are semi-conservative. Namely, if I have 2 demethylated strands and I replicate, half the methyl marks are going to be here and maybe the other half are going to be here. And then when I generate a new strand, the fact that half of the newly formed DNA has the old DNA modification from pre-replication means that there are enzymes that go in and establish the other half. And you know, it's a little more complicated for histone modifications because as you sort of divide the DNA, you have to push them off, but maybe the ones that are pushed off are sitting still nearby and they can be reincorporated, or they can be pushed over the replication fork, or maybe all the factors that are readers are also writers. So there's some kind of aspect of semi-conservativeness in epigenetic markers. Gosh, that was a long answer. Did you guys feel that you learned stuff? Okay, good. Yes. Both, mom and dad. Sperm has some modifications as well. Egg has some modifications. So yes, moreover, the environment of the sperm has a lot of additional stuff. So basically it's a full ginormous cell. And there's all kinds of small RNAs that could be regulatory, that could be then controlling all of that information, and so on and so forth. So yes, there's a lot of sort of maternal environment that contributes a lot to the offspring's epigenomic imprinting, or, you know, imprinting is a whole other thing of sort of parent of origin kind of effects, and sort of remembering the state that is deposited by the parents, and so on and so forth. So huge field. There's entire semesters worth of lectures on this. So any other questions on basic understanding? I mean, these are awesome questions, and I'd love to sort of chat a lot more. Okay, great. So here we are. We have 3 different types of epigenomic modifications that allow the cell to remember what it is. You know, if you're a liver cell, you don't want to deal with all of the stuff about, you know, neurons. If you're a plumber, you don't want to deal with all of the stuff about painting or about ballet or about nuclear physics. You just want to care about your profession. So in the same way, every one of our cells wants to care about its profession, and that's how we need to sort of remember that state, and that's how epigenomic information, that's what epigenomic information does. But you might ask, how does a liver cell decide to turn on all the programs that make it a liver cell, right? So that's what we're going to be talking about today. So basically, there's at some point reading of the DNA sequence to establish the active state or the inactive state, okay? There's regulators, control regions, that depending on the cell type that they're in, decide what information should be utilized and what information should be hidden for every one of the cell types in your body. These regulators bind in promoter regions and in enhancer regions. Promoter regions are the proximal regions to where transcription starts and RNA is made and genes are utilized. Enhancer regions loop around to bind those promoter regions and recruit the RNA polymerase and the RNA making machinery to effectively lead to this generation of RNA and ultimately protein. Okay, so these enhancers and these promoters have different epigenomic marks, and part of the reason they have different epigenomic marks is that they have different sequence properties that recruit different regulators. Okay, so in the DNA itself lie a sequence of patterns, a sequence of motifs that guide the binding of these regulators. And of course, the cis regulatory machinery, like the stuff that's on the same side from the Latin, you know, cis, and then the trans machinery, the stuff that's from across, from the Latin trans, which means across, collaborate to make those decisions. So I might have the nicest landing site for, I don't know, GATA1. Like I made the most beautiful binding site. GATA1 is not expressed. Is it going to bind? No, right? So that's sort of the transacting component of gene regulation. So you have the cis-acting, which basically tells you what are all the motifs in a particular location. And then the transacting tells you what are the proteins that are actually expressed that can recognize these sequences in that cell type. And that's how liver cells end up with a different epigenomic landscape than brain cells, because all the liver factors were expressed in that cell type. Raise your hands if you're with me. Awesome. Your mind should be hurting if you're really understanding me, because now you might be asking, well, who's deciding the transacting regulators, right? And well, the transacting regulators are decided based on the epigenomic state of those. And oh, who decides the epigenomic state of that? Oh, well, the cis. And who decides the cis? Oh, the trans. And who decides that trans? And so on and so forth. Okay. So if you're really with me, you should be like really puzzled as to how is this even possible. And that's going back to the, I challenge any one of you to build an automaton that will actually develop into a human body. So yes, there's a lot of chicken and egg kind of thing, where you basically have initially an environment established in the zygote that will then lead to asymmetric cell divisions that will establish the basic germ layers of endoderm, ectoderm, mesoderm, eventually, you know, neuronal lineages that will then differentiate further. And after every one of those, you're establishing part of the transmachinery prior to this asymmetric cell division. And then you continue establishing the cis prior to the next division and so on and so forth. Okay? Is everybody with me as to sort of how you're resolving this chicken and egg problem? By sort of consistently establishing asymmetries during those divisions. And not everything is completely cell autonomous. There's a lot of signals that are deposited by the mother during gestation and during sort of the initial rounds of cell division that basically say, hey, this is where you should put a head, and this is where you should put the legs, and this is where you should put the front, and this is where you should put the back. So anterior, posterior, dorsal, ventral axis of patterning play a major role in Drosophila, for example, but also a role in many other species, including human. Everybody with me? All right, so now, how is this done? How are these transcription factors binding the DNA? Well, they recognize the particular set of atoms that every one of these nucleotides has, okay? We like to think of them as TAATTA, or the TATA binding motif, TATA binding protein, TBP, super creatively named. Mine's the TATA motif. You can see why it's called that. And the way that it's done is it doesn't have a sheet where it reads TAA. It basically senses the atoms that are making up these letters. So the A base, for example, has, you know, a particular set of atoms that stick out that even if you don't open up DNA, you can feel from the outside. These bases are stacked on top of each other, and then the proteins go off and touch them and feel them, and that's how you end up with this binding. Basically, the protein has evolved into a set of complementary shapes that will basically recognize a particular sequence sitting on the DNA at any particular location. So they have what we call an affinity for that sequence. So some of them have an affinity first to DNA, so then they can sort of bind DNA and then scroll around. And then they have an affinity for a particular sequence of DNA, which means that as they're scrolling, when they see that sequence, they're going to like to stick around it. Everybody with me so far? And depending on the particular structure of that protein, it recognizes different sequences. Okay? Bonus points. Who can tell me what's funny about this sequence? See one hand up? Someone can reverse complement DNA in their head? I want to see a few more hands. I kind of gave away the answer. What's funny about the word Anna, which is the name of my mother, and also the middle name of my daughter? It's palindromic, right? So I can read it forward, Anna, and backward, Anna, right? The same way, I can read this forward and backward, and it's the same thing. You're like looking at me weird. It's not the same thing. It is the same thing because you're reading DNA from the 5' to the 3' direction, and it's self-complementary. Every time you see a C, you have a G here, a T, CG, etc. So CAG, GTG on the opposite strand is exactly CAC, GTG. Okay, it's what we call a reverse palindrome, right? Everybody with me so far? And then, you know, GATA is a very famous transcription factor, as I mentioned earlier, and then the TATA box, etc. Everybody with me here so far? Awesome. So you end up with basic words in the DNA that are recognized by these proteins and that then bind to establish the transcription of particular genes and also of transcription factors that are acting in trans into other regulatory loci and so on and so forth. So then the cycle continues. So I can turn on genes, these genes can turn on other genes, and so on and so forth. Everybody with me so far? Awesome. So now mutations, of course, can happen, and then these might disrupt the binding of a protein. Why is this important? If you look at genome-wide association studies, these are studies of common genetic variants that are, you know, sort of with us for tens of thousands of years and that are pervasive in the human population. These common genetic variants are underlying a massive number of disorders. There's 120,000 genetic associations with complex traits in human disease, scattered across the genome. 120,000, this is mind-boggling. Any one of them might sort of increase or decrease your risk for one or many diseases in some particular case. Turns out that from those 120,000 common variants that are associated with disease from genome-wide association studies, from those, 93% do not affect the proteins. 93%, okay? So the vast majority of genetic variation associated with complex traits is sitting in non-coding regions. So part of the reason why we care to understand genetic variation, and in particular why we care to understand non-coding region circuitry, is because if we can understand how is this C going to lead to, I don't know, schizophrenia or Alzheimer's or obesity, then we can be one step closer to actually developing therapeutics that can counter those effects in living, breathing human people, including your friends, your family, etc. Everybody with me here so far? So basically these DNA nucleotide alterations, 93% of them are non-coding, and those underlie the vast majority of these complex traits that sort of make us different from each other. So there's very different structures, they recognize very different motifs, and we can represent those motifs by basically summing up information across all of the binding occurrences of a particular transcription factor. If we take here, for example, the ABF1 transcription factor. Why are they called transcription factors? Because they multiply transcription by a factor of 2 or a factor of 0.5 and so forth. Okay, so they basically alter transcription. So that's why they're called transcription factors or regulators or TFs. Okay, so the transcription factor sequence specificity can be inferred by lining up experimentally all of the locations where that factor was found to be bound. Okay, so ABF1 was bound in all of those locations. And if you add up the counts of A, C, G, T at each of those locations, you end up with a logo or a motif or a position weight matrix that basically tell you with what frequency is each of the nucleotides appearing in each of those positions. Raise your hands if you're with me. Awesome. Great. So those motifs are basically the workhorse of gene regulation. They are what establishes the cis circuitry to which the transacting factors will bind to then turn on and off different chromatin states, different gene regulatory programs, and so on. Everybody with me? So when we talk about a motif, we talk about a structure that cross-cuts the genome. This is something that is found hundreds or thousands of places in the genome. So the motif is basically this logo here. It's not, you know, a sequence. It's a generative model that can make sequences. You can sample from that model. It's kind of like giving you a Gaussian for the height distribution of the class rather than telling you the height of one person. You can sample from that Gaussian to get the height of individual people, but the Gaussian is the motif. It's the distribution over sequences. Raise your hands if you're with me. Awesome. So that's the motif. The motif instance is any one instance of that. It's any one sample. It's the height of any one student. Okay, everybody with me? Awesome. And that makes a bunch of assumptions. The first assumption is independence. The fact that I have an A here does not bother me at all if I put a G or an A here. That kind of model, position weight matrix model, assumes independence. The second one is that there's a fixed spacing. If this is a variable gap, I just have no way of encoding in the PWM. Okay, and there are more complex models. For example, I could have a set of PWMs. One set could be with an A here and an A here to encode that dependency. Another set could be with a gap of 4 or a gap of 5 in this particular location. Okay, ready with me? Any questions? Yeah.
Just to clarify, so this means that this is switching factors,
Yeah. You're basically what you're noticing is, wait a minute, 6 nucleotides? That's not a lot. That's going to happen a lot of time. So once every 4 to the 6 nucleotides, which is tiny compared to 3 billion nucleotides in human genome. You're absolutely right. So a lot of the binding happens combinatorially by basically having many transcription factors bind together with their motifs. So a lot of what we're going to learn today is how do we build models that can recognize not just the individual motifs, but also the combinations of motifs that make up higher order structures like grammars or sentences or, you know, paragraphs of gene regulation. That's a great question. Other questions? Yeah.
Just what the last row in the table means.
So there's a shorthand, because when you're typing up text, like when you're texting your friend who have this awesome motif, it's very hard to sort of write that motif in text. We don't have an emoji yet for every single combination of motifs. So if you want to text your best friend the best motif that you just found, you can basically use a one-letter shorthand. R stands for A or G, purine. Y stands for C or T, pyrimidine. S stands for strong. C or G is S because it has 3 hydrogen bonds. A or T is W for weak because it only has 2 hydrogen bonds. And then the other ones are H for just not G, and D for not C, and so on and so forth. And N for any. Okay, so that's sort of, if you want to text your friends, your favorite motifs. No texting during class. Wait for afterwards. I know it's important. All right. Any other questions, comments? All right. So there's motifs everywhere. So I talked about enhancers and promoters, but there's also post-transcriptional motifs. There's splicing motifs. There's exonic motifs. There's translational motifs. There's localization motifs that basically export the RNA and put in a particular organelle. There's motifs that sort of control the degradation and the lifespan of an RNA molecule. There's motifs that control the binding of microRNAs to sort of prevent translation or to lead to degradation. There's motifs that control both whether you're going to increase or decrease the frequency with which an exon will be included, and so on and so forth. Okay, so motifs are everywhere. So everything you're going to learn today applies extremely broadly throughout gene regulation. And as I mentioned, these motifs can be at the core of disease. In this particular case, in work that we did in my lab in collaboration with many other groups, we basically looked at this FTO locus, which is the strongest genetic association with obesity. And we found that out of this massive, massive association, we could trace it down to a single nucleotide alteration in this AT-rich interacting domain that changes a T into a C. And when this motif is no longer AT-rich, this AT-rich interacting domain protein, ARID5B, again, very creatively named, can no longer bind this AT-rich motif because now has a T in the middle. And that regulator normally represses these genes, which we found out are master regulators of a switch inside your fat cells that decides whether you're going to be storing the calories that you did not use today as fat, these lipid-rich molecules that sort of are inside your fat cells, lipids, are storing all these excess calories in your diet that you did not burn sort of chasing tigers and being chased by saber-tooths, etc. All of these extra calories are now going to be stored and make, I don't know, America obese and the world. Or they can be burned inside your fat cells and dissipate heat. That process, that switch, is controlled by these 2 regulators, which are controlled by this repressor, which binds a single nucleotide alteration, which can then lead to an increase or a decreased risk for obesity. I'm homozygous risk for this. Thank you, mom and dad. So I cannot burn calories, so I have to really watch my diet. So I've been starving since yesterday before giving this lecture in order to look decent for you guys. Anyway, my wife is homozygous non-risk, so she can eat anything she wants. It's crazy. So 40% of European chromosomes carry the risk variant, only 5% of African chromosomes, so suggesting perhaps a selection for, I don't know, dealing with the ice age when you're not sort of eating at McDonald's, but instead you might go a few days before you kill your next mammoth. And this is now sort of one potential therapeutic avenue for combating obesity by changing not just what you eat and whether you exercise or not, but the third equation of any thermodynamic equation: energy in, energy out, energy lost. And the energy lost is heat. Okay, so basically you can control now one more knob to sort of make people healthy. So that's the basics of gene regulation. It's humongously important and it's extremely complex, but at the root lie this very simple framework of combinations of regulatory motifs recruiting together transcription factors and establishing epigenomic landscape. Raise your hands if you're with me so far. Awesome, great. So this is sort of a whole module that we're going to be dealing with gene regulation. So I think it's important to sort of conceptualize all that early on. So now, how do we probe gene regulation? How do we even begin? Well, one workhorse of gene regulation is something known as chromatin immunoprecipitation, followed by sequencing. This was much cuter when we didn't have massively parallel sequencing and we were hybridizing to a microarray known as a chip, because it was ChIP-seq, which is kind of cute. But anyway, ChIP-seq is what replaced ChIP-seq. And it's basically chromatin immunoprecipitation. What the heck does that mean? Well, chromatin, you all know what it means now. It's sort of condensed stuff that contains both proteins, as in histones, and together they make up nucleosomes, and DNA. So chromatin is the combination of DNA and histones that it wraps around. So chromatin we understand. Immunoprecipitation. Well, precipitation we understand. When it precipitates, it rains, right? Don't tell your friends, ooh, it's starting to precipitate. So anyway, precipitation basically means falling down, okay? Again, pre and then zip, so on and so forth. Immuno means you're using an antibody to precipitate, to basically bring stuff down. So immunoprecipitation basically means I'm going to use an antibody to bring down chromatin, okay? So that's what ChIP means. How do you make these antibodies? Well, you basically make antibodies by feeding a bunch of innocent goats and bunnies, super cute animals, a bunch of the protein that you care about. So if you basically care about the ABF1 regulator, you basically feed the bunny all the ABF1 regulator you get. You can feed it. The rabbit is like, I hate that stuff. It basically builds a bunch of antibodies, and then you, the rabbit, and you take its blood, and you basically use these antibodies inside the blood to basically be able to pull down the ABF1 regulator and wherever it's bound in the genome. Is everybody with me? I won't play this lecture to my kids. All right, and now that you've pulled down all of these regions, you can basically ask, well, what is the DNA that came along with these transcription factors? Okay, so I can basically do a ChIP-seq experiment for a particular regulator and pull down all of the regions that are bound by that regulator. Okay, I can then sort of reverse the cross-linking that I had previously put in between chromatin and DNA to basically be able to sequence or hybridize or do anything else with those those sequence segments, those DNA segments, and then I sequence them and I map them to the genome, and I have all of the places in the genome where that particular regulator was bound. Raise your hands if you're with me. Awesome, great. So I can do that with a particular antibody against a regulator, a transcription factor, or I could do that for a particular modification. So if I want to find out all the regions that have H3K4ME3, I just put a bunch of H3K4ME3 histones inside, the little bunny, and then I sort of get an antibody that's specific to that modification. Everybody with me? So I can do that systematically and basically map out all the regions that are bound by different regulators or all the regions that have particular histone modifications. Okay? So yeah, you basically start out with protein and DNA together, you chop it up, you first cross-link it, then you chop it up, and then you pull it down, and then you reverse the cross-linking, and then you sequence and you map to the genome, and that's how you end up with something that looks like that. That basically says, when I did my sequencing experiment, gazillions of sequences fell here, and very, very few sequences fell there. So I can basically infer the locations of binding of a particular regulator, or I can infer the locations where a particular histone modification is found. And if you look closely here, this is H3K4ME1, mark of enhancers, H3K4ME3, mark of promoter, and then H3K36 trimethylation, mark of transcription. So you basically have an enhancer here surrounding the transcription start site, a promoter that basically says here's where action is going to happen and it's unidirectional, and then you have transcription. Ready with me? So that's one way of basically probing where's a particular regulator going to bind. But you can also probe simply all of the locations where DNA is accessible, where chromatin is accessible, where transcription factors could basically go in and access. Okay? So how do you do that? By basically using an enzyme, DNase, that basically eats up DNA. It sort of goes and cuts DNA. Wherever it's accessible, it just cuts it. Wherever there are nucleosomes, it can't access it, so DNA remains intact. And then you sequence, where did I get cutting? Where did I get all the fragments? And you end up with a profile of cutting on the DNA itself that basically tell you that there was no DNA accessibility here and there, which basically means that nucleosomes were covering that sequence, which basically means that that sequence was closed. And you have massive accessibility in between the nucleosomes. Raise your hands if you're with me on this one. Awesome. Now, what's really, really cool is that a transcription factor that is bound to the DNA will basically create a little dip inside a peak of DNA accessibility. So at low resolution, you basically see these peaks of DNA accessibility. At super, super high resolution, you see peaks and dips within the peaks. Everybody with me? So then we can kind of infer that something's going on in that piece of DNA and that it is regulatory. We have no idea who's binding there, but we know that, you know, it's accessible, okay? And then if we want to guess who might be binding there, we can look within the overall region of accessibility at low resolution and search for motifs, or we could look within the footprint in the same way that we saw for ABF1. We could basically map all the locations where it's bound, look at its footprint, and then infer what are the sequences within there. And that's how you can find out the transcription factor. Everybody with me so far? Awesome. So this is DNA digestion that basically sort of uses this enzyme DNase. And there's another way of probing accessibility, which is basically using transposase. So transposons are these mobile selfish elements that basically replicate within our DNA. And sort of they hop around and they can transpose. They can basically put themselves in other places. From the Latin trans and pose, which means put across. So this is assay for transposase accessible chromatin, or ATAC-seq. It's pronounced attack because you're actually attacking the genome, okay? Some people say ATAC, that's fine too, but ATAC is how it was originally intended to be pronounced. Everybody with me? So ATAC-seq basically attacks the DNA with a bunch of transposases, and the transposases basically insert themselves, and then you basically figure out where did the transposase insertion events happen, and that's how you figure out accessible DNA. Everybody with me? And you can now basically sort of figure out, well, first of all, how much they agree. There's some disagreement between them. Some of that comes from the inherent specificity of DNAs to basically bind particular locations and so on and so forth. And you can also play a trick, which is to look for forward and reverse reads on the DNA. And the forward reads are basically telling you that, ooh, there's all these DNA segments that I captured with my chromatin precipitation. I'm going to sequence the beginning of those segments, but the stuff that I pulled down must be to the right of that. And then the reverse reads are basically telling you, ooh, I captured something, it ends here, but it goes on for a little bit. And then that should be sort of pointing to the left. So if you have a bunch of reads pointing to the right and a bunch of reads pointing to the left, it kind of tells you that the actual binding event must have been in the middle. Everybody with me? So that's sort of, you know, one way that you can get higher resolution. So you can also find very distinct profiles of binding associated with different regulators. And the reason for that is that the chromatin landscape is shaped by these factors in very particular ways. And there's, you know, all kinds of sort of binding events that are preventing this accessibility. And you can kind of infer where are the nucleosomes positioned, where are the transcription factors bound, and so on and so forth. So basically you have nucleosome, accessibility, nucleosome, accessibility, binding of transcription factor, accessibility, nucleosome, accessibility, and so on and so forth. Everybody with me? So there's a lot of information in those experiments, yeah.
So what would be the axis of this graph?
The x-axis is the genomic axis, and the y-axis is amount of DNA accessibility, just number of reads. Great question. Any other questions?
If you compare ATAC-seq and DNase-seq, which one is more similar?
It's a complicated answer. So basically, you know, it's a complicated answer. I mean, DNase is usually, you know, higher resolution and higher quality, but it's a lot more expensive. So a lot of people are doing ATAC instead. But there's all kinds of information that ATAC captures, which in some cases is similar to MNAs, which basically tells you where nucleosomes are positioned, etc. So there's advantages and disadvantages to both assays. The main advantage of ATAC is cost. So people are doing a lot more ATAC nowadays. And John Stam is doing as much DNase as the rest of the field is doing ATAC. Okay, anyone else? All right, so basically the goal is perhaps regulatory motifs can predict DNA binding. Maybe I can sort of say, oh, if I can sort of see where are these motifs happening in the genome, I might basically have a rational way of understanding the makeup of these gene regulatory regions. And that's what we're going to try to do. Basically, we're going to try to sort of infer the words that make up gene regulation in this lecture, okay? So this is known as regulatory genomics. So regulatory means gene regulation, that means control of the genes, and then genomics means that we're gonna do this at large scale across the genome, okay? So what are some of the traditional ways before deep learning for studying gene regulation? Well, one of them is to basically look for co-regulated genes. You basically take a bunch of genes that are turning on and turning off in the same conditions. For example, you can look at liver. What are all the cells that are expressed there? Neurons. What are all the cells that are all the genes that are expressed there? And then look at the promoter regions of all these genes to basically find motifs that might be indicative of liver function and indicative of brain function. Everybody with me? So basically one way is to look for co-regulated or functionally related sets of genes, and then look for common patterns upstream of them, common motifs upstream of them. So you could basically align the promoters to each other using some kind of local alignment and use some expert knowledge perhaps for what the motifs should look like, and maybe find a median string by enumeration, or look within evolutionarily conserved blocks, and so on and so forth. And what you end up with is a twofold problem, right? If I knew what the motif looks like, I could basically search for that pattern in each of the sequences, and then I would know the starting positions. If conversely, I knew the starting positions where that transcription factor might be bound, I can just put them on top of each other, stack them, just like we saw for ABF1, and then infer the motif, okay? So we have, you know, a variable that's missing here, which is where are all the starting positions, and another variable that's missing, which is what is the sequence pattern of all of those motifs, okay? When we have two variables that are both missing, either one of which could help inform the other, we basically carry out in traditional machine learning a procedure known as expectation maximization, which basically means that I can repeatedly use one to estimate the other and use the other to maximize the you know, overall probability of the sequence occurrences of the first, okay? So expectation maximization, okay? And that's sort of the traditional approach to motif discovery. We basically start with a lot of locations, a lot of regions of the DNA where you know motifs might be enriched within them. And then you basically infer a motif from them by basically guessing what might the motif be, and then guessing what might the starting positions be, and then guessing what might the motif be, and doing this repeatedly until you basically converge to some kind of pattern of sequence that is overrepresented. This sounds like magic. If I start with crap, I'm going to end up with crap in the next iteration, right? So if this is a completely nonsensical sequence and that's a completely nonsensical sequence, how could I possibly ever actually converge on the motif? Would be obviously your question. And the answer is that because you assume that the motif is enriched, you have slightly higher chance of landing on an actual motif site than you have on landing on a completely unrelated site. And that sort of works to your advantage. So after a few iterations, you end up with more of your starting positions falling where the enriched motifs are. Is everybody with me here on sort of the intuition behind expectation maximization, or EM, and then the intuition as to why would it even work? Sounds good? Any questions so far? Yeah.
Is this done probabilistically, so that you have some chance of escaping the local minimum? Or
Absolutely. So there's many ways of doing this iteration. One of them is in this maximization step of, in this expectation step, after you've computed what is the expectation for all of the, every starting position of containing that motif, you can then say, ooh, I'm going to choose the maximum of sort of that vector of annotations. That's one approach. That's called the greedy approach, because every single time you just take the maximum. Another approach is to do what's known as Gibbs sampling. And Gibbs sampling basically uses that probability to sample one location from that probability distribution. So if that has a probability of, I don't know, a score of 8, and that has a score of 6, and that has a score of 3, with, you know, a small fraction, I'm going to be picking that 3, and only that 3. And with some higher probability, I'm going to be picking the 8, and only the 8, and ignoring the other. So that's Gibbs sampling. And again, it's stochastic. It's sampling from the posterior distribution of the distribution that you're trying to approximate. There's another approach, which is expectation maximization, which is using an integration over all starting positions, each weighed by its probability. So if I have a 0.8, a 0.6, and a 0.3, I'm going to average them together, weighted by their scores. Does that answer your question? Any other questions? Again, every question has an, you know, informative answer that sort of everyone's going to benefit from. So never hesitate to ask questions. Yeah.
Yeah, I was wondering if you could motivate again the reason why you use expectation maximization. I think it came out of some of the limitations of enrichment-based discovery.
Yeah. So the idea here is that you're trying to find a common sequence pattern, an enriched sequence pattern. And one way to do that would be to count all k-mers. The problem is that the k-mer that you're looking for might never actually occur there, because motifs might be degenerate. They might bind with mismatches. So the reason why we're doing this repetitive way is because that allows us to estimate not perfect sequence matches, but approximate sequence matches. And that's sort of the workhorse of all of these motif discovery approaches. The fact that we can discover these common patterns de novo. Any other questions? Yeah. No, even without knowing it, your scores are approximating it, and then you're refining it at every step. As I'm refining my motif, the posterior distribution of the true answer is getting closer, basically is getting closer and closer to the true answer. But initially, it's just an approximation of where are these motifs actually occurring. So Gibbs sampling allows you to basically sample from a distribution you don't have by sort of constantly re-estimating that distribution. Any other questions? Okay, awesome. So that's sort of the key idea behind traditional motif discovery, and there's many technologies for inferring these motifs experimentally, and there's many ways of inferring these motifs computationally: expectation maximization, Gibbs sampling, enumeration, correlations with peak height, evolutionary conservation, learning about protein domains, etc. So there's many ways of doing this traditionally. What we're going to focus on today is how do we use deep learning for doing this systematically. Okay. So what are we going to use from deep learning? What we're going to use first is convolutional neural networks. So what are convolutional neural networks? The whole concept of convolutional neural networks is that you can learn these intermediate representations at every step of the way. For images, we saw how you could initially hard code your image patterns and then use them to basically recognize motifs. So what we're going to do here is, I'm looking for an overview slide. Should have added one. What we're going to do here is use the same concept of, at the lowest level, I'm basically capturing individual representations. And then I'm combining them into higher order representations and higher order representations, and eventually predicting the affinity of a regulator binding them. Okay, so In the same exact way that for images, we had these convolutional filters that were looking across the R, the G, and the B of an image, the RGB, the red, green, blue channels of an image, and then learning patterns which are recurrent throughout the image. And remember, with convolutional neural networks, I basically had this set of parameters that I was reusing all the time. I don't know if you remember that, but who remembers that? I had actually put them here, so they're still here, cool. But anyway, so basically I had these fixed set of sort of convolutional filters that I was reusing over and over again. And one convolutional filter was recognizing edges pointing that way, another one sort of little circles, and so on and so forth. And then at the next layer up, I had another set of convolutional filters that were recognizing maybe, I don't know, the claw of a cat and the eye of a human and the wheel of a bus and so on and so forth, okay? So in the same way here, we're gonna learn layers of information. We're gonna learn convolutional layers of combinations of the primary sequence, the motifs that are learned from the sequence, combinations of those motifs, higher order structures, and so on and so forth. Raise your hands if you're like, wow, this is kind of cool. Yeah, awesome. So basically, this whole methodology that we sort of have already developed of learning these convolutional filters from the sequence itself, we're now going to turn around and apply them to DNA sequence. And there's two ways to do this, right? One is to hard code the filters. Basically, learn the motifs from somewhere. I don't know, pay a bunch of people to just go and figure out every motif, and then combine them in a hard-coded way, right? So that's sort of the old way of doing things, where you have feature engineering, where I'm sort of discovering the best possible motifs. And then all the system can do is basically figure out how to combine these motifs together in super interesting ways using the fully connected layer. Another way, the more modern way, would be you let the system learn the set of convolutions directly from the problem at hand. So it learns the set of filters rather than just hard coding them, and then it gets to reuse them. So there's parameter sharing across many, many different locations in the genome where you're going to be reusing the same small set of filters. Raise your hands if you're with me on this one. Awesome. Any questions? Yeah.
First off, like I can understand the car, like different layers of abstraction.
Yeah.
What exactly is the problem?
Yeah, so excellent question. So basically, at the lowest level, you have DNA sequence. Right above that, you have motifs, ABF1, GATA, etc. Then ARID5V. Then above that, you have combinations of those motifs. If I have this motif and that motif, maybe that would be one combination of motifs. Remember how, someone had pointed out that, hey, these motifs must be occurring everywhere. I think it was you. You basically said, oh, that means that they're very common. And the reason for that is that there's only so much information in the occurrence of any one motif, but combinations of motifs carry a lot more information. So maybe one of my convolutional layers will basically start learning combinations of those motifs. Does that answer your question? Any other questions? Yeah.
Is there, so what we're trying to predict is the affinity of those rather than gene expression itself. Is there a biological reason for expecting that different combinations of motifs would affect the direct binding interaction of the protein with the DNA?
It's a great question. So basically, yeah, of course. So there's two things that you can predict. The first is you could predict the binding of a single transcription factor, okay? But the much more interesting thing to do is to maybe predict the DNA accessibility of a particular DNA segment. So if I'm trying to predict DNA accessibility, then there's a whole set of convolutional filters that I want for overall DNA accessibility in a particular cell type. And these filters are basically all of the regulators that might be active in that cell type. Whereas if I only have one experiment of one transcription factor, chances are that the main motif that I'm going to find is going to be the transcription factor. Not the only one, and the reason is that there's a lot of combinatorial binding. that whenever this transcription factor occurs, chances are its buddy is also there because it doesn't like to bind alone. It's feeling lonely, it doesn't have anyone to chat with. So there are protein interaction domains that basically help refine the specificity of a particular transcription factor. And everywhere you see its motif, yes, it's going to be attracted, but it would rather only stay at those places that also have a binding site for its best friends. Does that make sense? So basically, very often, even in the binding of one regulator, you will end up finding multiple motifs for its closest friends. Any other? Yeah.
So when you say feature engineering, you can do process using domain knowledge to label motifs.
For example, if I know that this transcription factor is a helix loop helix transcription factor, or if I know that this transcription factor is a homodimer, like we saw with the reverse palindrome, then I might engineer constraints into the set of features that are driven by the biological knowledge. That would be feature engineering. I might say, well, hey, don't worry about it. I know the set of motifs. These are the motifs that I'm going to use. Or I might say, well, I know the types of motifs and the family of structures that I expect. Or you might say, just let me use some experimental approach to find all the motifs, and then this is going to be my set of filters, convolutional filters, and I'm going to only use those. Or you could just say, let's just discover the motifs de novo. Does that answer your question? Any other questions? All right. So, so basically what we're going to do is infer a set of filters, a set of convolutional filters that are going to be reading the DNA sequence and then interpreting as motifs and then combining these filters together into higher order structures, okay? And then these convolutional filters, the same way that we scanned our image with these orientation or circle, etc. detectors, we're going to be scanning our sequence with these convolutional filters. So the convolutional filters are basically going to be some type of representation of the underlying motifs. And scanning the sequence basically means that I'm going to be scoring that sequence for occurrences of that motif. And in the same way that we used max and sum and other sort of pooling layers, now we can use these layers to basically say, hey, did that motif occur? Or did a motif occur? And so on and so forth. So basically we're going to use a max or an average or a sum to basically bring information that becomes independent of the exact starting position. You could basically have pooling layer with, 20 nucleotides underneath, basically says, I don't care exactly where in those 20 nucleotides this motif occurred. I just care that it occurred. Sounds good? So again, the way that we're going to be scanning DNA sequence is in the same way that we had the RGB values of every pixel. Basically, when you look super, super close at your monitor, you basically see that there's actually 3 different lights. There's a red light, a green light, and a blue light. And your brain is sort of averaging them together to sort of create a color. In the same way, there's different channels that you can use to basically understand images. And in the DNA example, those different channels are going to be one hot encodings of each of the letters. So if I have 4 DNA letters, A, C, G, or T, what I'm going to use is a vector that is basically 0 everywhere except where there's an A, and a vector that's 0 everywhere except when there's a T, and a vector that's 0 everywhere except where there's a G. And every single time, that vector is going to light up on my original sequence, okay? So this is how I'm encoding my DNA sequence. I basically have, A, T, G, C, A, G, C, etc. Sequence, I encode it as 4 vectors, each of which is a binary vector. And at every position, I have 4 channels, A, C, G, or T. And then the particular nucleotide encoded is whichever channel is 1 at that location. So my convolutional filter is going to be looking at these 4 channels simultaneously and then lighting up exactly when the right nucleotide appears in the right channel. Raise your hands if you're with me. Awesome. Very cool. All right, so what we saw earlier were these position weight matrices that basically tells you with what frequency do I see each of the nucleotides. And we drew a logo for these sequences based on the information content of that position. If I tell you that I see a G in that location, how much information did I give you? Well, if you knew nothing before, you now have 2 bits of information because it takes 2 yes/no questions and answers to infer what nucleotide it is. First, I can ask, well, is it a purine or a pyrimidine? You can say, well, yes or no. Yes, it's a purine. And if I know that it's a purine, I can say, okay, now is it an A or a G? Therefore, every letter, because it's one of 4 options, gives me 2 bits of information. Everybody with me here? Now, this only gives me 1 bit of information. It says, well, it's G or C, right? So that's why I plot it with a height of 1. So the more I specify something, the more information I get. And the maximum information I can get is from 0, where basically all 4 bases are equally likely, to 2, which is 2 bits of information. If the background nucleotide composition is AT rich, I don't start at 0. I sort of kind of know that I'm expecting an A or a T. So the information content of seeing an A might actually be less than 2. Because, yeah, I was kind of expecting that. But if it's a G, it might actually be more than 2. Because, wow, I'm really surprised. I was not expecting that. Everybody with me? So basically, biased nucleotide content means that this is actually skewed. So anyway, this is, you know, just the basic position weight matrix. But in practice, what I'm actually using is a position-specific scoring matrix that basically can have positive scores for the characters that I care about and negative scores for the characters I don't care about. Everybody with me on the difference between a position weight matrix and position-specific scoring matrix? So the PSSM is basically asking what is the probability of observing the sequence divided by the probability of that particular nucleotide being generated by a background composition. And if my background composition is, I don't know, AT rich, then this is basically saying, oh, that's much more likely with a background. Everybody with me? So I can basically now scan my sequence pattern across the DNA and basically score in the same convolution operation that I did for images. I can score a DNA sequence using my first filter. And I can score it again using my second filter, using my third filter. So I have this whole stash of filters that I can now scan across the sequence and score the, the evidence, if you wish, that this particular motif occurs at that particular sequence. So if my motif is, I don't know, GC, this particular here, which basically has, C or G, and then really strong G, really strong A, really strong T, really strong A, and then, kind of weak A. So GATA. I'm now scoring GATA across my sequence. So what am I going to get? I'm going to get a positive score here with a G, I'm matching, and a negative score here with a C, and a negative score there, and so on and so forth. And that's at the first location. At the second location, I'm now doing a little better. So basically here, I have G, C, A, and so on and so forth. It's not quite GATA, but it's a little closer. And I can get basically, a slightly better score. And then as I scan my GATA sequence using this convolutional filter across these 4 vectors of one hot encoding of DNA sequence, at every single step, I'm basically doing a convolution of the 0-1 sequence with the particular position PSSM, a position-specific scoring matrix, and I'm getting a single number as a score. Raise your hands if you're 100% with me on this one. Yeah? Awesome. Raise your hands if you have any questions. Yeah, go ahead.
Why don't we do something like softmax for the scoring weights here?
Yeah, so you can do that right after. So basically, this is just a very basic bottom layer. Then you can do all kinds of additional operations on top of that. So I'm going to get there soon. Next question.
For instance, G might be
Yeah, so you could use a combination of filters. One filter basically says A must occur with A, and the other filter says it's the same exact motif, but G must occur with G. So instead of having a single convolutional filter that captures the dependency, you split your motif up into two convolutional filters that basically score for one version or the other version of that dependency. Does that answer your question? Back here and then here. But now we're going to learn that matrix. So basically, yes, I could use it using a bunch of previously known motifs that I've learned from elsewhere, or I could learn the convolutional filters themselves. So we're going to actually learn them. Yeah.
So the sequence is that only the like one hot encoding ones. You can actually have the quality score.
One way to encode quality score might be to reduce that one hot vector to basically, instead of having 1, make it be 0.7, because I'm kind of uncertain about that base. And instead of having 0, 0, 0, I could put 0.1 for all of the other ones. So the quality score could be encoded there, or you could have a separate channel that basically tells you about the quality score. And then you might scale everything based on that quality score channel to basically decrease the match accuracy of something in a location that has a lower quality score. So yeah, you could use quality scores within that encoding. Other questions? Awesome. You could also use evolutionary conservation. You could basically say, well, across 29 different species of mammals, here's the evolutionarily conserved sequence. Instead of having a one hot encoding that only has zeros and ones, you could have what is the consensus sequence of that alignment. Or can I have an additional track that basically tells me is it conserved or not? And that could actually boost up your accuracy score if indeed motifs are evolutionarily conserved more often than non-motifs. Okay, awesome. So basically, we have the makings of a convolutional neural network for regulatory sequences. So basically, you can learn about motif combinations by sort of having these pooling layers that look for the combination of orange, blue, and green, regardless of where they might be. So if I have a sort of pooled layer at a resolution of 50, for example, then that pooled layer could basically say, yes, I did see all three of them co-occur. Or it might say, oh, yes, I saw these co-occur much more closely. Or, yes, I saw 3 copies of that regulator, and so on and so forth. Okay? You can then start building these classifiers and basically start asking what is the linear combination of these parameters. And that's where you sort of introduce your nonlinearity right after. You can basically say, great, I have now added these scores, and now I'm going to have a thresholding function that basically tells me between 0 and 1, did I actually see the score? And for example, you could use a sigmoid or a logistic function that basically can allow you to infer the probability that something occurred. Or you could use a rectified linear unit, which can basically say, well, if it's below some threshold, I don't care. But after that threshold, I want to actually be notified of the actual scale and the motif match score of that location. So you basically can use these artificial neurons to now represent these motifs and then scan them across your sequence, okay? So here's the overall architecture. Basically at the bottom, I have my DNA sequence. Then I transform into a one hot encoding, which basically has this 4-dimensional input at every location. And then I have these convolutional filters that are sort of reused. It's a common set of parameters that I sort of bring in each time. And this is where I can learn these parameters at the same time that I'm learning how to predict my sequence. And then I can basically scan with each quote unquote motif or each convolutional filter, and I get a different score for each filter. That basically tells me, oh, this motif occurred in these locations, and that motif occurred in those locations. And then after that, I can have basically a thresholding layer that basically uses ReLU or sigmoid or something else. And then I can have these convolutional filters at the top, which are basically pooling all of this underneath, all of this information underneath. And that can basically give me the label of the class that I'm trying to predict. So now, putting it all together, I could basically have all of these bound regions have a class label of bound. And all of these unbound regions have a class label of unbound. And I'm trying to learn how to classify bound from unbound. And I'm basically doing backpropagation, and I'm learning both what are the filters themselves and where should those filters be, you know, where all those filters are occurring, where are those instances, these high scoring motif instances from my overall motifs. So basically, number one, I'm learning these shared parameters, and number two, I'm learning the position specific parameters that basically tell me where are all those occurring actually. Everybody with me? So at the end, I can basically make a single prediction that basically tells me, hey, is this bound? Or I could actually make a multi-task prediction that basically tells me, is this bound? And is that bound? And is that other factor bound? And so on and so forth, okay? And there's many architectures that have been developed for deep learning with regulatory genomics. One of them is DeepBind, that basically was using a set of known convolutional filters from independent experiments, and then sort of first inferring these filters as one step, and then combining them into another step. And sort of, you know, you basically take the current batch of inputs, you basically convolve it, you have your scan of each of those motifs, basically have these, you know, ReLU layers and pool layers, and then you feed all that into a fully connected network that then allows you to predict whether something's bound or not, or a particular transcription factor is binding. And then you use those prediction errors to update the weights and maybe even update the motifs. Is everybody with me on this architecture? So that's sort of DeepBind. And it, sure enough, was able to discover a lot of motifs that were, you know, matching the known specificities. And it was then able to use this as a mutational map that basically says, if I change that T2C nucleotide in this AT-rich motif, how likely is it that the binding prediction will actually change? So you can basically use this convolutional network that you've learned to then scan both the reference sequence and your alternate sequence, which might be disease associated, to basically say, did I predict a change in the specificity, or a change in the affinity, or a change in the accessibility, or a change in the binding, and so on and so forth? Raise your hands if you're with me. Okay. So that's sort of the first such publication. So Ali Panahi et al. In 2015 from Brenda Frey's group. The second application was DeepSeq, that was basically directly trying to predict from the DNA sequence the mutations that are disease associated. So this was basically predicting features from the reference allele and for the alternate allele in one shot in order to sort of predict the impact of gene regulatory mutations in non-coding regions. So rather than learning a model only on the reference and then seeing where that score changes, you were directly trying to predict those changes. Everybody with me? And then maybe, you know, the most vanilla one, if you wish, the most perhaps elegant one is Bassett, that was developed here at the Broad by David Kelly and John Wren's group. That was basically learning those convolutional filters in what would be the class hallmark convolutional neural network. So basically, you're simultaneously predicting DNA sites in 164 different cell types. So you're sharing these convolutional filters across different cell types, and then you're applying them to this multitask prediction challenge, where you're basically saying, well, a small set of regulators are constantly reused across all of the different cell types of the body. Let me now learn what are these convolutional filters that I should be using, and then use that to predict jointly the accessibility of all of the different cell types. Raise your hands if you're with me on this one. Awesome, great. And then the motifs discovered again matched many of the known regulators sort of for those sequences. So, I don't know if we have too much time for questions. Maybe we can talk right afterwards. And then Computer was recently developed by Anshul Kundaji's group. And then what they're trying to predict is not simultaneously multitask across different cell types, but more simultaneous multitask across different transcription factor binding events. And the idea is that you're going to be reusing your motifs, your learned convolutional filters across many tasks simultaneously. Okay. So that's what I want to leave you with. Basically, this Computer, basically you can have, you know, much more complex architectures. You don't have to just have sequences input. You could also have information about DNA accessibility, about smoothing, about the actual conservation, and so on and so forth. And then you can achieve very good performance there. And then yeah. So very quick question. Go ahead. Oh, well, it's much better than an SVM. So here's some performance differences, and you can sort of see, how much would a random forest do, how much does computer actually do, and so on and so forth. So basically, there's a dramatic increase in accuracy. And as the task that we're giving these regulatory models is increasing in complexity, I think, a lot of the traditional approaches of PWMs and, random forests and all these regression models are vastly outcompeted by these deep learning models. Who feels that they've learned stuff today? Awesome. See you guys on Thursday.